Concept 15.3 focuses on linked genes. So what exactly is a linked gene? Linked genes are genes or a gene that are located near each other on the same chromosome and tend to be inherited together in genetic crosses. This example may help you to better understand what linked genes are. Here you can see that A and B are in fact linked genes because they are on the same chromosome. However, A and C are not linked because they are not on the same chromosome. In other words, any two genes found on the same chromosome are said to be linked. To see how linkage between genes affects the inheritance of two different characters, let's examine another of Morgan's experiments.
A guy by the name of Morgan designed an experiment to show how linkage between genes can affect the inheritance of two different characteristics. He used flies in his experiment. He had wild-type flies, which had gray bodies, and we can represent that with a B+. And those wild-type flies also had normal wings, and we represent that with a VG+. And he crossed those wild-type flies with a mutant type of fly, which had black bodies that we represent as just a B, and short wings, which we represent as just a VG. These traits, the mutant traits, are both recessive to the dominant traits. The P generation would be shown as B plus B plus VG plus VG plus times the mutant type, which is BBVGVG. After you cross those, you would move down to the F1 generation, which the results were a dihybrid female gray body normal winged fly, which that means it would be B plus B VG plus VG. And then they cross that with another mutant fly, which would be represented as BBVGVG. Now this one, the normal type is female and the mutant is male. And that is always because the recessive genes are always on the male. After that, you get your test cross results, which ends up in 4 different eggs, which the first one is B plus VG plus, which means gray bodies and normal wings. After that, you get BVG, black bodies, short wings, B plus VG, gray bodies and short wings, and then BVG plus. And then over here you have the sperm, which I said earlier, it's always the male, it's always the mutant traits, which is BVG, black bodies and short wings. After you cross all of those, you would have these different crosses, and the first one is a wild type, the normal wild type, which is gray and normal. The next one is the black and short wings, so it is black and short winged. The next type you get is gray and short. And the last type you get is black and normal winged. And you are left with 965 gray and normal to 944 black and short winged 206 gray and short, and then 185 black and normal. Some of the predicted ratios that Morgan thought he was going to get is that if genes are located on different chromosomes, he would get a 1:1:1:1 ratio. And if genes are located on the same chromosome and parental alleles are always inherited together, he would get a 1:1:0:0 ratio. And these are the ratios that he got. This concludes that since most offspring had a parental phenotype, which means 965 of them did, Morgan concluded that genes for body color and wing size are genetically linked on the same chromosome. However, the production of relatively small number of offspring with non-parental phenotypes, which are these over here, indicated that some mechanisms occasionally breaks the linkage between specific alleles of generations on the same chromosome.
Okay, this video is going to explain the recombination of unlinked genes. Through crosses of two different characters with different traits, Mendel learned that some offspring of these parents have traits that don't match either parent. So by doing a cross similar to this one with a heterozygous dominant parent and a homozygous recessive parent, Mendel learned that 50% of the offspring will have the same phenotype and traits as one of the parents, while the other 50% will have a new phenotype with new traits that don't match either of the parents. The phenotype that is the same as the parents is referred to as a parental type, and this new phenotype that 50% of the offspring will have that is not similar or the same as either of the parents, highlighted in red, is known as a recombinant type, or recombinants for short. Geneticists learn from these kinds of crosses that 50% of the frequency will end up being recombinants. Now, this can only happen if the genes are unlinked. So in order for 50% to end up parental types and 50% to end up recombinants, making it a 50% frequency, the genes must be unlinked. And the recombination of these unlinked genes occurs during metaphase 1 of meiosis and is random. This causes the independent assortment of the two unlinked genes.
The next concept regarding linked genes is called crossing over. Now, you may recall that crossing over occurs in prophase of metaphase 1 and involves an exchange of information among homologous pairs. So in crossing over, homologs come together and go from something like this to something like this, and then they separate and each have new information on them. This means that offspring don't always share the same genotype as one of their parents for a linked gene.
New combinations of alleles, variation for natural selection. First, we need to go back to review what we already know to understand how this works. In Chapter 13, physical behavior of chromosomes during meiosis contributes to the generation of variation in offspring. Each pair of homologous chromosomes line up independently of their pairs during metaphase 1 and crossing over prior to that during prophase 1 can mix and match parts of maternal and paternal homologs. In Chapter 14, Mendel's experiments show that the behavior of the abstract entities known as alleles of genes also leads to the variation of offspring. By putting these ideas together, you can see that the recombinant chromosomes result from the crossing over may bring alleles together in new combinations, and the subsequent events in meiosis distribute the gametes, the recombinant chromosomes, in multiple combinations. Random fertilization then increases the number of variant alleles combinations that can be created. This abundance of genetic variation provides the raw genetic material on which natural selection works. If traits conferred by particular combination of alleles are better suited for a given environment, organisms possessing these genotypes will be expected to thrive and leave more offspring ensuring the continuation of their species. The next generation of alleles will be shuffled anew. Ultimately, the interplay between environment and genotype will determine which genetic combinations persist over time.